بسم الله الرحمن الرحيم، والصلاة والسلام على خير خلقه محمد وعلى آل بيته الطيبين الطاهرين. اللهم صل على محمد وآل محمد وعجل فرجه. The title of this lecture is Microbial Nutrition and Nutritional Requirement, prepared by DR.IHSAN DR.IHSAN from Department of Microbiology, College of Medicine, University of Basra. The objective of this lecture is define microbial nutrition, 2. Determine the type of nutrients, 3. Classify the microorganisms depend on the type of nutrition, 4. Discuss the importance of each nutrient in the living microorganism, 5. Clarify physiochemical factors affecting on microorganism life. What is a nutrition? Nutrition is a process by which a chemical substances, that mean nutrients, are required from the environment and used for cellular activities. That is mean of essential nutrient that must be provided to an organisms. Two categories of essential nutrients found in media or in environment, either micronutrients or micronutrient or trace element. Macronutrients require in large quantities, rule in cell structure and metabolism, example protein and carbohydrate, while micronutrient or trace element are required in small amounts, involved in enzyme function and maintenance of protein structure, such as manganese, zinc, nickel, etc. Also may be divided nutrients according to chemical structure into inorganic nutrients and organic nutrients. Inorganic nutrients like atom or molecule that contain a combination of atoms other than carbon and hydrogen, such as metal and their salt, such as manganese sulfate, ferric nitrate, sodium phosphate, and gases like oxygen and carbon dioxide and water that belong in organic nutrient division, while organic nutrients contain a carbon and hydrogen atom and are usually the product of living things, such as a methane, CH4, carbohydrate, lipid, protein, and the nucleic acid. In the bacterial body or bacterial cell, there are 70% of water and a protein. 96% of the cell is composed of 6 essential elements: carbon, nitrogen, oxygen, hydrogen, phosphorus, and silver, and the percent appear in this table. The requirement of growth may be divided into two requirements, either physical requirement or chemical requirement. The physical requirement is temperature, pH, oxygen requirement, osmotic pressure, and hydrostatic pressure and radiation, the latest two factors affecting on growth physically on growth of bacteria, while chemical requirement is found of carbon, oxygen, nitrogen, silver, phosphorus, trace element, and organic growth factor. What mean of organic growth factor? Growth factor is organic compound that cannot be synthesized by an organism and must be provided as a nutrient, such as essential amino acid, vitamin, nitrogen base, other. Like in Haemophilus influenza, must be provide factor X and Y to maintain the growth of bacteria on a specialized media. Now, let's study essential organic material that affecting on growth. Will be start by carbon. According to carbon, we can classify the bacteria into heterotroph and autotroph. Heterotroph organism that obtains carbon in an organic form made by other living organism: protein, carbohydrate, lipid, and nucleic acid. While autotroph is an organism that uses CO2, an inorganic gas, as it is carbon source, not dependent on other living things. So we can classify the microorganism according to carbon source and energy source into the following form or following group. The nutritional type, chemo, meaning a chemical compound, while a nutrition type, photo, meaning a sunlight. We can classify into four groups. First group is a photoautotroph, or unknown as generally as a autotroph, that is take a carbon source from CO2 and energy source from sunlight. The second group is chemoautotroph, that take a carbon source from CO2 and energy source from simple inorganic chemical. The third group is a photoheterotroph, that take a carbon source from organic material and energy source from sunlight. While the finally group is a chemoheterotroph or heterotroph, that the pathogenic bacteria belong on this group, that take both a carbon source and energy source from organic compound. Other heterotroph that not belong any of fourth groups is a saprops. Saprops decompose a dead organism and recycle element, release enzyme to digest material. And parasite. Parasite utilize a tissue and fluid of living host and cause a harm or disease. The environmental influence on microbial growth is a temperature, oxygen requirement, pH, osmotic pressure, radiation, and hydrostatic pressure. Firstly, temperature. According to temperature degree, we can classify the microorganism into three groups. Psychrophile, the first group, psychrophile, that the optimum temperature below the 15 centigrade and capable of growth at 0 centigrade. Example of this psychrophile is Erythrobacter species, Psychrobacter species, and member of the genera Halomonas, Pseudomonas, Hyphomonas, and Sphingomonas. The second group is a mesophile, that the optimum temperature is between 20 to 40 centigrade. Most of human pathogens belong to this group. The third group is a thermophile, that the optimum temperature greater than 45 centigrade. That is, good example is a Thermus aquaticus that grow in hot spring. It was about 90 centigrade. What is the physiological adaptation habit in that halophiles and that thermophile, because to become a resist of high temperature. What is the modification happen in thermophiles? Firstly, relative greater concentration of guanine and cytosine in DNA and RNA. Guanine and cytosine base pair involve in a 3 hydrogen bond, making them more stable at high temperature than adenine-thymine base pair that form only a 2 hydrogen bond. Thirdly, stability of DNA and RNA is critical to cell survival. Fourth, enzymes thermally stable. Fifth, high concentration of hydrophobic amino acid. Six, cytoplasmic membrane is modified. And 7, high proportion of high molecular weight saturated fatty acid. The second element affecting on growth is oxygen requirement. The molecular oxygen O2 as a necessity of life, but it is actually in a sense of poisoning gas. Metabolic system require oxygen for aerobic respiration, and microbes that use molecular oxygen mean aerobes produce more energy from a nutrient that microbe that cannot use oxygen and known as anaerobe. Superoxide, dismutase, catalase, and hydrogen peroxidase that cleavage the H2O2 resulting from found or presence of oxygen and water and yield H2O2, that is a poisoning chemicals for the living organism, and this enzyme may be a catalyst or maybe break down the H2O2 and yield H2O and oxygen free. Enzyme which neutralize toxic oxygen. According to oxygen requirement, we can classify the bacteria into 2 main groups, aerobes and anaerobes. And below aerobes, there are many divisions. Aerobes mean require oxygen, that's obligate aerobes, cannot grow without oxygen, and facultative anaerobe, that capable of growth in the absence or presence of oxygen, and the third group is a microaerophiles. This group does not grow at normal atmospheric tension of oxygen, such as bacteria grow in a soil, water, or in the human body, like Helicobacter pylori, Campylobacter, and Borrelia. Helicobacter pylori cause a gastric ulcer. The second type of group that divide according to oxygen requirement is anaerobe, that does not require oxygen, like a Clostridium, because not have any of the catalyst enzyme. There are another group classified according to this element, a capnophiles. Capnophiles need a higher concentration of CO2, like a bacteria known as Spilothrix. And finally, a group of bacteria, it is aerotolerant anaerobe. Aerotolerant anaerobes does not utilize oxygen but can survive and grow to limited extent in its presence, like a Streptococcus and bacteroid. We can grow anaerobic bacteria in this instrument known as anaerobic jar. Thus, maybe must be yield anaerobic condition inside the growth environment. We use a gas pack. When put water in this pack, that yield or that produce a hydrogen. Hydrogen will react with oxygen and yield a water, and the CO2 may be resulted inside of this anaerobic jar. There are anaerobic indicator strip put here in an anaerobic jar, that is a methylene blue, become colorless and absent of oxygen and kept of blue color when found or when presence of oxygen. The third factor affecting on growth is pH, and according to pH we can classify the bacteria into two main groups. Alkylophiles, that the optimum pH is relatively to highly basic, such as Bacillus okinensis and Alkalibacteria perennis. The second group is a neutrophil, the optimum pH range about pH 7 plus or minus, that means the human pathogen, all human pathogen or most of human pathogen belong to the neutrophil. The third group is acidophile, that the optimum pH is relatively to highly acidic. Example, Acidobacterium or Thiobacillus, or most of fungi grow in acidic condition. The fourth factor affecting on growth is osmotic pressure. Microorganisms obtain almost all their nutrients in solution from the surrounding water. Osmotic pressure, water diffusing across the cell membrane in the response of salt concentration. The bacteria having 70 to 80% of water require water to grow. A group of bacteria that is live in adverse condition are known as xerophiles. Xerophiles are extremophilic organisms that cannot grow and reproduce in condition with a low availability of water. Xerophiles are often said to be xerotolerant, meaning tolerant of a dry condition. They can survive in a nutrient with water activity below 0.8, like endoliths and halophils are often xerotolerant, like a trichosporidiosis nigrinensis. Extreme or obligate halophils adapted and require a high salt concentration, like a halococcus or halobacterium. The fifth factor affecting physically on bacterial growth is a radiation. There are two types of radiation, either a short wavelength high energy radiation or a large wavelength low energy radiation. The good example for the short wavelength is a X-ray or gamma ray, while a large wavelength like a UV light. There are some bacteria is a radio of the microorganism that capable of living with a high level of ionizing radiation, like bacteria appear here and known as a Deinococcus radiodurans. The radiation are great for killing the bacteria and used for sterilization of bacteria. According to damage of DNA, make a thymine dimer in the double strand of DNA in sufficient quantity that can kill the microorganism, and in lower range because a mutagenesis, make a mutation, cause a melanoma or xeroderma pigmentosa. The spores tend to be resistant to radiation and can survive much longer exposure. The final factor affecting on growth is a barophile, and known as also a piezophile, is an organism which survives at a high temperature, such as a deep sea bacteria or archaea. They can generally found in ocean floors, where the pressure often exceeds to 380 atmospheric pressure. Bacteria that grow at moderately high hydrostatic pressure in ocean. So we can divide the bacteria into three groups according to the level of hydrostatic pressure. Parotolerant grow at a pressure from 100 to 500 atmospheric pressure, like Bacillus subtilis. Barophile grow at pressure of 400 to 500 atmospheric pressure and grow from 100 meter to 1 kilometer like a xenophytophores, while extreme barophilic bacteria that able to grow in higher than 500 atmospheric pressure like a Halomonas cellular that able to grow from 1000 meter to 9000 meter. Thank you very much for your listening.